Hey guys, good afternoon. So in this lecture I'm going to go ahead and cover several things regarding animal specialized systems. So these come from chapters 40 through 43. In later videos I'm going to go ahead and cover the rest of the animal systems because there is a lot to talk about here. Normally this would take about 3 to 4 weeks to cover in class, but due to the current situation, evidently I gotta break it up into two long lectures. So the first one on animal specialized systems, I'm gonna go ahead and talk about osmoregulation, nutrition, digestion, gas exchange, animal nervous systems. So in osmoregulation, I'll be differentiating between how different animals keep a homeostatic balance of water and electrolytes in their bodies. And then nutrition, digestion, I'll go ahead and talk about the digestive systems of animals. And then gas exchange deals with the pulmonary system, the lungs. And then animal nervous system, I'll get into the central nervous system of upper animals, like for example the brain and the spinal cord and the nerves. Okay, so again, I'll cover all the details and in two videos. This is video number 1. So let's start off with chapter 40, which is on water and electrolyte balance in animals, otherwise known as osmoregulation. So we have animals that live in water, those are aquatic animals, and we have animals that live on land, these are terrestrial animals. The difference is pretty evident in their ecosystems. Obviously, the aquatic animals live in water and need to come out of the water for temporarily for a little bit of time to carry on with some functions that they cannot necessarily do under the water. And terrestrial animals spend most of their time outside of the water. They live under atmospheric pressure within a biosphere which is full of gases. So there's the different mechanisms which they use to in order to be able to keep their balance of water inside of their bodies, which is different from animals that live, exclusively inside of water. So here we're looking at an osmoregulator that lives in seawater. Seawater contains a high degree of sodium chloride. Sodium chloride is known as salt. It's the table salt that we consume. And what's important about table salt is that it contains electrolytes. It contains ions. Okay, electrolytes and ions are essentially the same thing. So salt is made up of sodium and chloride, as a lot of you know. So what fish need to do, they have one important adaptation that allows them to keep their water regulation inside their bodies as opposed to outside of their bodies. They're called gills. So you've heard of gills before. This is a zoom in of gills. Here we're looking at an animation here inside of the gills. Here's the inside space of the gills. This is the seawater, the external space, and they're separated by membranes. Okay, so these are membranes right here. All right, so now notice the red dots right there. Those red dots, those represent the concentration of ions inside and out of the cells of the gills. Okay, so outside the gills in the seawater, there's a higher concentration of sodium and chloride, and these are solutes. Why are they solutes? Well, because they're ions that are dissolved in water. Okay, so now the concentration of sodium chloride outside of the gills in the water is higher than the one inside of the gills, gill cells. Alright, so now keep in mind that in order to move ions in water into and out of the cell, all animals, and even plants for that matter, all animals need to use a combination of the different cell transport processes that we have covered in the past. And I'm referring to diffusion, osmosis, active, passive transport, and so the concentration of salt in seawater is said to be hyperosmotic due to the fact that this water contains a higher concentration of salt than the inside of aquatic animal cells. Thus, again, so these are the different processes that are required to carry on with osmoregulation in all animals. Okay, so they would be passive transport. Keep in mind from the beginning of the year, passive transport has different types. Okay, first is we have simple diffusion, facilitated diffusion, and there's another one not shown here. It's called osmosis. Okay, so simple diffusion does not require any protein, any membrane protein. These molecules are small enough that they can diffuse, they can move right through the membrane, and the way in which they move, that is basically why it's called diffusion. Right? Diffusion allows for molecules to move from areas of higher concentrations to areas of lower concentrations until a balance is achieved, and that balance is homeostasis. At least in terms of electrolytes and molecules. Now, facilitated diffusion requires a protein. This is a membrane protein that spans the entire membrane, and this protein has a channel that allows molecules to move from the outside to the inside or the inside to the outside, depending on the concentrations, again from high to low. Now the other one is called osmosis. The other passive transport type is called osmosis. Osmosis uses a pore. It's a membrane protein. It's called aquaporin. Okay, so aquaporins allow water molecules to move into and out of the cell depending on the concentrations as well. Water will move from areas of high concentration to areas of low concentration until balance is achieved. Now, active transport is called active because it takes energy. Okay, the biological word for energy is adenosine triphosphate. Okay, so it's actually the molecule. I should have said the molecule. The biological molecule for energy in biological systems is called adenosine triphosphate, or ATP. So ATP is an important regulator of, all, if not all, mechanisms. Most, if not all, mechanisms within cells. Okay, most of them require energy. So if it does require energy, then ATP is going to be involved in that process. How does that differ from passive? Well, in the fact that molecules are going to move from areas of low concentration to high concentration. Okay, not like in passive where it's the other way around. So these molecules are going to tend to be electrolytes. Again, electrolytes are ions like sodium, potassium, chloride, magnesium. So if they are small molecules and they're ions and they contain a charge, then they're going to be electrolytes. Okay, electrolytes allow for gradients to be made in the membrane of cells. Gradients are important because they drive cell transport processes, and they're also important in, for example, nervous systems where electrical potentials need to be made in order for the nervous systems to work. Okay, so those are just a few examples out of the many. So different marine species maintain osmolarity in the different manners. I'm showing two. One is osmoregulation, the other one is osmoconformation. Okay, so in osmoregulation, these animals are going to go ahead and maintain a tissue osmolarity that is different from the environment. Okay, so it's going to be lower than the environment. For example, here we're looking at the osmolarity of seawater, and osmolarity is essentially a measure of the concentration of salts in water. Okay, so the osmolarity here in seawater is 1,000 milliosmoles per liter, and the osmolarity inside of the cells of this fish is 410. Okay, so again, osmolarity in the cells of osmoregulators needs to be maintained lower than that of the outside. Okay, so now in osmoconformer species, they need to maintain a tissue osmolarity that is either close to or equal to that of the environment. So for example, this shark here has cell osmolarities of 1,000 milliosmoles per liter, whereas which is the same as that of seawater. Okay, so again, these are called osmoconformers, and these are called osmoregulators. Again, many different types of osmoregulation. These are two of the most important and significant ones. Now, these are not perfect mechanisms. Okay, there's trade-offs that need to occur. For example, in osmoregulators, there's a trade-off, biological trade-off between gas exchange and osmoregularity. Okay, so what does that mean? Well, that means that while they can keep osmoregulation balanced, that right there is going to go ahead and cause another disbalance in other molecules. For example, here urea concentration in these sharks is going to be a lot higher than the urea concentration of osmoregulators. Okay, so that right there, urea, by the way, is a waste product of protein synthesis. Okay, protein synthesis, the making of proteins in cells, and the breakdown of proteins puts out some byproducts in the form of urea and uric acid. Okay, so this right here is going to call for other adaptations to be able to deal with the urea concentration, which I'll get into in a bit. So if salt concentration is in the osmoconformers goes higher than the outside salt concentration, or higher than the normal, then sharks are going to go ahead and utilize an organ called the rectal gland, which they're going to go ahead and use in order to get rid of extra sodium. The ways in which they do it is by using sodium potassium ATPases, which are membrane receptors or membrane proteins that work through gradients. Keep in mind, gradients are concentrations of ions on different sides of the membrane. Here's a membrane. This is one cell of the rectal gland in sharks. So there's different concentrations of sodium chloride and potassium outside and inside of the cell, thus calling for the generation of a gradient. Okay, so these gradients again are simply made by different concentrations of ions in cells versus outside of cells. So sodium potassium ATPases require ATP. That's first and foremost. These are energetic. These are energetically expensive, if you will, molecules here. So what they do is they go ahead and exchange sodium for potassium. Okay, so if the concentration of sodium in this cell, for example, is too high, then sodium potassium ATPases are going to go ahead and exchange potassium for sodium, in that manner lowering the amount of salt. Sodium chloride potassium co-transporters, they go ahead and move these three ions using the same membrane protein. Okay, so and again, it all depends on the concentrations of sodium, in this case sodium chloride and potassium. These are going to tend to move from to low concentration, but they can also be moved actively from low to high concentrations depending on what the cell needs. Potassium and chloride channels move potassium and chloride accordingly outside of the cell depending on the concentration, and then sodium is it's not only a small molecule, a small ion, so small that it can fit right through the cells to be able to be excreted out of the cell, but it's in such a high demand that it's easy for sharks to bring in too much sodium into their bodies. So in that case, this adaptation here allows the sharks to get rid of sodium to the outside from their bodies in order to be able to reach a homeostatic sodium concentration in their cells at rather rapidly, if you will. So terrestrial animals work a little bit differently. Okay, so let's start off with insects here real quick. Insects have this system called the tracheal system, which is equivalent to the trachea of animals or higher animals. Now, what this tracheal system does is it takes in carbon dioxide and oxygen from spiracles. So, in spiracles are openings located throughout the body of the insect, and that's how they're able to breathe in and out. They also have water inside, but the thing is, on the outside, the way in which they regulate water is by preventing the loss of water, because terrestrial animals do lose water to the environment because of the fact that there's sun, there's air that allows for evaporation. So insects have a chitin wax covering all around their body, and chitin is a carbohydrate. We're looking at a carbohydrate layer here. It's called chitin again. Here's the name. And so what chitin is going to do is going to go ahead and act as a cuticle that is adapted to prevent water loss from the body of the insect. Now, in osmoregulating, insects are going to go ahead and use the equivalent to a kidney, right? Which is called a Malpighian tubule. So this Malpighian tubule is connected directly to the gut of the insect. Okay, so in this Malpighian tubule, this is a place where they're going to go ahead and exchange water and electrolytes, and they're going to go ahead and get rid of nitrogen as waste from the synthesis of proteins, the synthesis of nucleic acids, but also their breakdown. Okay, so the breakdown of proteins and DNA creates these molecules in the form of urea and uric acid. Those are toxic for animals, so therefore we need to get rid of them because we produce them. We produce them as well. Now, insects get rid of them by using Malpighian tubules. They diffuse right into the Malpighian tubules, or they can also be exchanged with the different proteins that I talked about earlier. And then from there, they're routed over to the gut of the insect, and then finally excreted in the form of urine or feces. On higher animals like mammals, for example, and vertebrates for that matter, we have a urinary system. Okay, so the urinary system is driven by kidneys. So these are bean-shaped organs located retroperitoneal. Okay, so peritoneal means the abdominal cavity, and retro means behind. So the peritoneal cavity is covered by a rather large membrane. Okay, so behind that membrane are the kidneys are located behind the membrane, and as well as the arteries and veins. So this artery right here is called the abdominal aorta, which branches into the renal artery that feeds the kidneys with oxygenated blood. Okay, so here's the left renal kidney, and this is the right renal kidney. Artery, I'm sorry, in red. Now the blue right here, this is the inferior vena cava, which reroutes all of the blood that came into the kidney. It's after it's used by the cells, after the cells have extracted the oxygen from it, then they go ahead and diffuse carbon dioxide from the cells right into the vein right here, renal vein, and then inferior vena cava, so that blood can be taken back to the lungs to be oxygenated again. So here's why the kidneys are important. Kidneys are osmoregulators. Okay, so this is the osmoregulatory organ of mammals. So kidneys contain three major parts. They have a cortex, which is the superficial layer right here. They have the medulla, which is the middle layer, and they have the pelvis, which is the deeper layer. From the kidney, urine is made. So urine is filtrated, if you will, from the blood. Once it's made, it goes ahead and drains into the ureter, which is right here. Right? So the ureter runs, it connects the kidney to the bladder, and it drains the newly made urine from the kidney to the bladder. Okay, the bladder is a storage site for urine, which has an inferior tube here called the urethra. The urethra extends into the genitals, and we use the urethra to go ahead and expel, to eliminate urine. Okay, so let's talk about the kidney real quick. So kidneys contain about, at least a human kidney contains about 1 million nephrons. Okay, these are nephrons right here. Animations of nephrons. Nephrons are located within renal pyramids, which are located in the medulla of the kidney. So here's the medulla again. The medulla is the middle layer right here. These are renal pyramids, I'm sorry. So here's 1, here's 2, here's 3. Inside of those renal pyramids are thousands of nephrons. Okay, these nephrons, at that point, nephrons are microscopic, so you need a microscope to be able to see them. And these are structures, again, that are specialized. They're adapted so that we can osmoregulate. Okay, so that we can keep our balance of water inside of the body at a homeostatic level, but also a balance of ions like sodium, potassium chloride at a homeostatic level inside of the body. There's a few parts to the nephron. Here's the first one here. This is the glomerulus. Okay, the glomerulus is the connection between the circulatory system. These are branches, if you will. These are branches of the renal artery, which they penetrate into the glomerulus, which is also known as the Bowman's capsule, and the renal corpuscle. So they penetrate in, they bring blood in, and then the glomerulus filters that blood. It filters it by using these slits. Okay, they're called fenestrations. Okay, here they are. So here's a zoom in into the glomerulus. These little lines that you see inside, those are called fenestrations. Okay, so they're made of specialized cells called podocytes that contain processes or extensions. Processes are extensions of podocytes, and these processes, again, they make these slits called fenestrations. That's how blood is filtered. So as blood comes in through the afferent arteriole, which is this right here. I'm sorry about that. So the afferent arteriole routes blood into the glomerulus. As it flows through the glomerulus, it's filtered using these fenestrations, these slits. So as it's filtered, it becomes filtrate. Okay, so this filtrate is ultimately going to become urine. And then the rest of the blood, once it's cleaned, if you will, it goes back into the efferent arterial, which is right here, and then back into the systemic circulation. And evidently by this point the blood is cleaner. Okay, so now in cleaning this filtrate, there's many processes that take place. Okay, so we're looking here at the proximal tubule. Let me take you back to the slide with the nephron. Here it is again. This was the glomerulus or the renal corpuscle. This space right here, continuous with the glomerulus, is called the proximal convoluted tubule. And then moving down, this is the descending limb of the loop of Henle. This is the ascending limb of the loop of Henle. And then this last part right here is called the distal tubule, or the distal convoluted tubule, which drains into a collecting duct, which is right here. Alright, so that collecting duct goes ahead and receives the filtered or the filtrate rather, which has been extracted from all of its waste. Okay, so all of its water, I'm sorry. So what's gonna happen is a few things. Let me go to the slide here. A few things are gonna happen. First and foremost, throughout the entire nephron, we're gonna have something. We're gonna undergo something called reabsorption, but we're also gonna do something else called secretion. So if we reabsorb everything that went into the filtrate, which is good for the body, is gonna go ahead and be reabsorbed back into the kidney. If that happens, then the kidney returns all of those good materials back into the blood and veins. Okay, so most of this reabsorbed material is going to be made up of water. So we need to be keeping as much water as possible. So we are going to reabsorb about 60-70% of the water that we put into the filtrate. Okay, so we reabsorb that water and then we put it back into the body by means of the arteries and veins of the kidney. Now, if we do have excess water, then we're going to go ahead and secrete it because it's in excess. Okay, so excess water is not necessarily good for the body because it dilutes the contents of cytosol, the cytoplasm of cells, which decreases concentrations, decreases gradients, and basically throws all homeostatic mechanisms off, which is not necessarily good for cells. I'm only showing you water here, but there's also the excretion and secretion and reabsorption of sodium, chloride, potassium, calcium, and so on. So all of these ions and electrolytes that are necessary for the body, depending on whether the body needs them or not. If the body doesn't need them, if we have excess, then we secrete them into the lumen of the nephron and then ultimately into the collecting duct, which leads into the renal pelvis, which leads into the ureters and then down into the bladder to be expelled. If we do need them, we are going to go ahead and reabsorb them. So all these processes that I just mentioned within the nephron, they're regulated by hormones. Okay, there's a few hormones of importance here. One of the most important ones is ADH. ADH stands for antidiuretic hormone. Diuretic, D-I-U-R-R-E-T-I-C. Sorry. So again, antidiuretic hormone is made in the pituitary gland within of the brain. So it's right below the hypothalamus, and it's released into the kidneys, into the blood, which eventually reaches the kidneys. So at the kidney level, ADH is going to insert aquaporins right there on the collecting ducts and the distal tubules of the nephrons, so that water can be reabsorbed. Okay, so anti means against, diuretic means diuresis or release of water. So what this hormone is going to do is it's going to prevent us from losing too much water. Now there's other hormones like aldosterone, for example. Aldosterone comes from the adrenal gland, which is located right above the kidneys, each one of our kidneys. So aldosterone plays a role in water volume, regulating water volume in our blood. Okay, so by doing that, it regulates our blood pressure, which also regulates our heart rate. Okay, so that's aldosterone right there. There's also another hormone which regulates water balance inside of the body. It's called ANP. Okay, so ANP stands for atrial natriuretic peptide. Atrial coming from the heart. Okay, so if you've taken biomedical science before, then you know a little bit of the heart. The heart has two major parts, okay, the atria and the ventricles. Atria is divided up into two chambers, and the ventricles are divided up into another two chambers. So that's 4 chambers for the mammalian heart. Now, the right atrium, the inside of the right atrium, the lining of it, is made up of endothelial cells. These cells produce ANP, or atrial natriuretic peptide, which plays a role in blood pressure. So the way in which it regulates blood pressure is by modifying the amount of water that is in the blood, and this takes place by either the constriction or dilation of blood vessels that are entering the glomerulus. Okay, so let me see if I can find, here it is. So these are the blood vessels from the renal artery that enter the glomerulus. So all these regulatory hormones are going to go ahead and have an impact on blood vessels, okay, whether directly or indirectly. They can either dilate or open up blood vessels. That's called vasodilation. If they dilate, then they decrease the peripheral resistance, which is going to decrease blood pressure. If they constrict, then they increase peripheral resistance, which increases in blood pressure. Okay, so the higher the blood pressure, the more filtration that goes on in the nephrons. Therefore, the more water we get rid of. Okay, the more water more water and electrolytes that we can excrete. Okay, but then the nephrons have their own local regulation here. They can either vasoconstrict this afferent arteriole. If they constrict it, then they reduce the amount of blood that goes into the glomerulus, therefore reducing the amount of filtration that goes on. So if you're very active, you're playing sports, well, your body doesn't necessarily need to be urinating at that point. Therefore, these afferent arterioles are going to constrict. Okay, so that's going to allow you to keep as much water as possible, not letting you filtrate, if you will, or producing urine. Alrighty, so let's talk about chapter 41 here real quick, animal nutrition and digestive systems. So let me start off by mentioning the stages of nutrition. Now keep in mind that in order for us to maintain nutrition, we need to ingest a combination of different nutrients, and these nutrients will be made up of proteins, carbohydrates, amino acids, enzymes, and lipids. Alright, so all of which are going to go ahead and help us do basically everything in the body, to include making proteins, to include making DNA, RNA, and carrying out with all of the functions of the cell. Now, in doing that, cells are also going to need other molecules like enzymes, for example, and vitamins and minerals for that matter. Okay, so those are going to be cofactors. They're going to be helpers of enzymes that are going to help cells in making ATP, so that cells can carry on with all of their processes here. So 4 stages of nutrition. First is we have ingestion. Okay, so putting food into our mouths, or what depending on this on the species that you're talking about, they have different variations of a mouth. All right. So first is ingestion. Second is digestion. So once that food makes it to the stomach, then the stomach starts breaking down the food and extracting nutrients, or rather for making it easier for the small intestine to extract nutrients. Then is absorption. So small intestines go ahead and absorb, at least invertebrates, go ahead and absorb nutrients from that broken down food. And then elimination. So whatever part of the food we don't need, we only extract nutrients, what's good for us, what we need, right? But the rest is waste. And so whatever we don't need, then we go ahead and put it into a large intestine, at least invertebrates, and then we get rid of it. And in this case, this animal uses the mouth for both, for ingesting and elimination. So what I'm showing here is the different important vitamins that our body needs, to include B vitamins, C vitamins, D vitamins, all of which need to be, taken in somehow, whether they're eaten or absorbed or made from UV radiation, like vitamin D. So what B vitamins are going to do is they're going to go ahead and take a very important role in the production of ATP. So making energy for cells. B vitamins are going to go ahead and take a role in that. Folate, or vitamin B9, aside from helping in making ATP, it's also going to help in making nucleic acids. Okay, so and proteins for that matter. So it's a very important role. It plays a very important role in the making of DNA, RNA, and also metabolism of amino acids, or breaking down proteins. Vitamin B12 is another coenzyme which is also involved in making DNA, nucleic acids. Vitamin C is important for the making of collagen. So collagen is super important. It plays a key role in all of our body systems, especially those that provide support and allow us to move. Okay, so for example, in tendons, ligaments, which we have thousands of, those are made up of collagen fibers. Okay, so vitamin Vitamin C, is involved in making that collagen inside of our cells, so that we can, maintain and repair tendons and ligaments rather easily. Vitamin D can be made from exposure to the sun. So that's naturally how we're supposed to make it. But we can also consume it. Almost all foods that are natural contain vitamin D3. If it's processed foods, then it's probably going to be enriched with some vitamin D, which is not necessarily a bad thing. But regardless of that point, most of the population of the U.S. Lacks vitamin D, is insufficient in vitamin D. Okay, so if there's no issues with you getting out in the sun, then go ahead and get out in the sun and enjoy some vitamin D that is began in synthesis by your skin, okay, by skin cells. But its synthesis, starts in the skin, and then the liver is a mediator of the molecules that are made in the skin. And then from the liver, it travels into the kidneys, where it's actually activated. Okay, so after it's activated by the kidneys, then vitamin D is going to go ahead and play a role in many, at least 300 to 400 different mechanisms. Okay, evidently I'm not going to mention all of them. But two types of mechanisms. One is absorption of calcium, which is a mineral, and other minerals at the small intestine level. And then the second major type of function is it plays a role in regulating genes. Okay, so it regulates a whole plethora of genes which are very active within the human body. Now, these here are minerals. Okay, these are metals essentially that we have to consume. So these so they play different roles in the body. For example, calcium and fluorine, they become part of teeth. Chlorine makes it helps generate gradients in cells to help in driving cellular mechanisms like cell transport. But it's also involved in the making of stomach acid, okay, HCl, which stands for hydrochloric acid. Iodine is important for thyroid function. The thyroid, which is located in the neck region, right below, right by the Adam's apple, which is called the thyroid cartilage. It produces hormones. Okay, these hormones are called thyroxine and triiodothyronine, which necessitate they need iodine to be able to be made. Iron is the last one here. Iron is very important for hemoglobin synthesis, which is located inside of red blood cells, and hemoglobin is important for carrying oxygen and carbon dioxide throughout the body. Alright, so here is the mammalian digestive system. Okay, so it all starts in the oral cavity, so the mouth right here. Then the pharynx larynx, which is right here. From there, it moves on to the esophagus, down to the stomach, the duodenum, which is right here. This C-shaped structure here is the beginning of the small intestine, and then the small intestine. So duodenum is the first part. The second part is the jejunum, and the third part is the ileum, which leads into the large intestine, which is right here. Okay, so here's the ascending colon, transverse colon, descending colon, rectum, and anus. So let's look at each one specifically. Here's the oral cavity, which contains a tongue. So teeth are important for obvious reasons. So we need to chew food. The anatomical word for chewing is masticating. Right, so we use teeth to be able to masticate food and break it down from bigger into smaller chunks. We produce saliva using salivary glands in the oral cavity. So food is mixed with saliva to make it easier for swallowing or deglutition and then digestion after that. The tongue is important. The tongue is a very highly muscular organ which pushes food back into the throat in order for us to be able to swallow that food. Now, once we have we've undergone deglutition, which is swallowing, then we go ahead and drive that food into the esophagus. Okay, what the esophagus is going to do is it's going to go ahead and begin rhythmic contractions, muscle contractions, because the esophagus is made up of smooth muscle. Okay, those contractions are called peristalsis, Peristalsis, peristalsis. So again, that peristalsis goes ahead and grabs onto the food, and then it, just by muscle contraction, pushes it down into the stomach. By this point, this food is not called food anymore, it's called bolus. Once bolus reaches the stomach, this is what happens, right? So here's the anatomical structure of the stomach. It's got muscle layers which are involved in churning. Churning is another variation of contraction. So the stomach's going to go ahead and churn and churn and churn. Is going to contract to be able to further break down the food into what's known as chyme. So once bolus has made it into the stomach, it's going to go ahead and be it's going to be squeezed, if you will, by the stomach lining, and at that point it's going to become chyme. So chyme is going to collect on the inferior part of the stomach, which is this part right here. This is the superior part of the stomach. This is the middle part of the stomach. And the connection between the esophagus and the stomach is called the gastroesophageal sphincter, which is a circular muscle right there. We have another sphincter located right around here. Okay, that's called a pyloric, Pyloric sphincter. Sphincters are important because we use them to limit transit, right? So if this sphincter is closed, it's not gonna let any stomach acid from entering the esophagus, right? So stomach acid naturally wants to come back and enter the esophagus. If that happens, that's evidently dangerous. Some of you guys have probably suffered from reflux before. That's it's pretty dangerous for the esophageal lining, right? Because it can dissolve those cells. Now the pyloric sphincter is going to prevent chyme from passing to the duodenum. Yes, we do want to achieve that, but we want to limit, right, the amount and the timing of chyme going into the small intestine, the duodenum, right here. Okay, so again, the stomach is gonna go ahead and churn. That's one, break food down. But it's also gonna go ahead and combine food or bolus and chyme at this point with stomach acid, okay, or gastric acids. And here's how that happens. It uses these glands here. These are called gastric glands. Okay, so these are located throughout the entirety of the lining of the stomach. These gastric glands are made up of different types of cells which produce mucus. Okay, so these are neck cells. They secrete mucus to keep the lining here protected from the acid. Okay, this is very thick mucus. Chief cells are located deep within the gastric gland, and then parietal cells are located even deeper than chief cells. Okay, so chief cells secrete something called pepsinogen, which must become pepsin once it touches hydrochloric acid. Okay, so these two are mutually dependent right here. One is needed by the other in order to function properly. Pepsin and hydrochloric acid are gastric juices which help in breaking down food further into what's known as chyme. Right, this is simply the mechanism of acid production. It happens by the use of hydrogen ions, which must diffuse energetically outside of these parietal cells. Now, there's different variations of a stomach. For example, here in this cow has a 4-chambered stomach with a rumen, reticulum, omasum, abomasum, which are different sites for digestion of plant materials here. Okay, so these animals are gonna contain they're gonna have the ability to digest and absorb nutrients from plant materials, which we don't necessarily have. Now, after the stomach is the location of the small intestine. Okay, so the small intestine is connected to the stomach here, and it's the same in humans, but the connection is different. But nevertheless, the function of the small intestine is the same. Here's the connection here. It's a C-shaped organ called the duodenum. Okay, so the duodenum is very important in the fact that it not only begins the absorption process of nutrients, but it also receives other materials from, for example, the pancreas and the liver. So the pancreas delivers bicarbonate ions to the duodenum, and the liver delivers bile to the duodenum. So the bile is used for the emulsification of fats, and then further the absorption of fats. Okay, for the absorption of fats, we have something called a lacteal. Okay, this green part right here is called a lacteal, and these lacteals are located inside of larger structures called villus or villi. Okay, so villi essentially line the entire inside lining, if you will, of the small intestine. So the inside of the small intestine is studded with villi. Inside villi, we have lacteals, which are important for fat and lipid absorption. Okay, so let's go ahead and jump over to gas exchange and a little bit of the circulatory system. Right now we'll go ahead and go into the details of this next week. Right now I'm going to go ahead and spend most of the time here on gas exchange. So why is the respiratory system important? Well, because it allows us to exchange gases. It allows us to get rid of CO2, which is toxic in high concentrations in our blood. And it allows us to respire, to breathe in oxygen. Why are those two important? Well, because oxygen is needed for mitochondria to be able to make ATP. Okay, it's something called aerobic respiration. If you forgot about it, go back to first semester cell respiration and read about it. So oxygen is collected inside the mitochondria to carry on with aerobic respiration, which is respiration, ATP production in the presence of oxygen. Okay, that's how we make most of our ATP. Now, that process puts out a waste product, many waste products, but one of them is CO2. So we need a little bit of CO2, and most of it is expired. So it's released into the atmosphere, and then as we breathe in and out, we exchange oxygen and CO2 through a respiratory surface. Okay, so this respiratory surface is essentially our entire respiratory system, which has different parts. So aquatic animals contain a set of gills. Okay, these gills are very important for aquatic animals because that is essentially their respiratory system. What gills are going to do for aquatic animals is through the use of something called a counter current exchange, these gills are going to go ahead and extract the oxygen from the water, and they're going to go ahead and get rid of CO2 from the metabolic mechanisms or metabolic processes that the cells of the fish underwent. So as the fish bring water in by drinking it, they go ahead and pass it through, right? They force it through their gills, and then their gills are going to go ahead and extract again the oxygen needed by the countercurrent exchange. So if water is flowing in that direction from left to right, then blood is going to flow in the opposite direction, and that right there is going to make up for the countercurrent exchange, which is going to make it a lot easier for gills to extract oxygen and also release CO2 into the water. So other animals use a spherical trachea system. Sphericals are openings, they're holes found on throughout the body of the animal that are able to bring in oxygen and put out CO2. These sphericals lead to an intrinsic and complex network of trachea which allow for respiratory exchange. Now the mammalian respiratory system is made up of a few structures. It's a bit more complex, starting with the nasal cavity, evidently in the nose, down to the nasopharynx, oropharynx, laryngopharynx, into the trachea, which is located within the thoracic cavity and is lined by cartilage, down into the middle of the thoracic cavity where it bifurcates into bronchi. Okay, these two tubes are called bronchi. Right here at this point, at the bifurcation or division, this point is called a carina. Okay, C-A-R-I-N-A. What the carina is going to do is important because it contains receptors. Okay, these receptors monitor the presence of debris that's not supposed to be there, like dust, for example. So if debris makes it into the trachea, it reaches the carina. It stimulates receptors that are gonna go ahead and trigger the cough reflex. So by coughing, we're able to expel all that debris that went in. What I'm saying is the only thing that should be inside of the trachea and the lungs is air, and that's it. Okay, air with oxygen and other gases, but the only relevant gas for us is oxygen. So as air enters the nose, nasal cavity, it goes through the nasopharynx, oropharynx, laryngopharynx, and then trachea, down into the bronchi, into the secondary bronchi, and then tertiary bronchi, which lead into these structures here called alveoli. Alveoli for plural, alveolus for one, for singular. These are crucial in exchange of gases because they do provide the respiratory membrane where gas exchange is gonna take place. So how is the exchange of gases gonna take place? So in our blood, as we are doing our everyday activities, our cells are active, right? So our cells are very active. The more active you are, the more the cells are active, and the more metabolism they're undergoing. If they're undergoing metabolism, that means that they're making ATP, they're making energy. If they're making energy, that means that they're putting out CO2. Where does the CO2 go? Well, it goes into the blood, right? So blood circulates using the circulatory system with a system of arteries and veins. So as blood collects CO2 and it passes through the lungs, it goes ahead and passes through the respiratory membrane and exchanges CO2 for oxygen to be able to take oxygen back into the cells. Now, how exactly does that occur? Well, using a set of muscles, bones, and cartilage systems here. First is this muscle here. This is called the diaphragm. Okay, so the diaphragm, when it contracts, it goes ahead and pulls on the lungs. It pulls them down to elongate this structure here called the thorax. Okay, so from the collarbone or clavicle all the way down to the end of the ribs, that's called the thorax. All right, that's your chest, if you will. So the diaphragm contracts, it pulls on the thorax, makes it longer for the reason that these lungs have to expand, right? So as you breathe in, the diaphragm is contracting, expanding the lungs, allowing you to bring air in to the lungs. So now as the diaphragm relaxes, then that puts pressure on the lungs, that squeezes the lungs basically, and that forces air out of the lungs. There's more to it, and I'll cover it in a little bit. Now, other muscles of respiration are located right between the ribs. Okay, so right between the ribs, those are called external and internal intercostal muscles. Right, so external, internal, intercostal muscles. So their contraction also helps in expanding the rib cage and lifting it, so that the space for lung expansion can be optimized. Okay, so the more space for respiration and expansion of the lungs, then the more air that can be brought in and the more oxygen that can be brought in. Now, there's more to that. Let me cover just one more aspect of it, and this aspect is dealing with pressures. So there's three pressures that are involved in respiration. One is the atmospheric pressure, which is provided for by the gases of the atmosphere. Okay, that's the atmospheric pressure. Now the intrapulmonary pressure is that pressure inside of the lungs, in the alveoli of the lungs. That's called the intrapulmonary pressure. Another pressure is called the intrapleural, P-L-E-U-R-A-L, intrapleural pressure. That is the pressure in the membrane of the lungs that surrounds the lungs and protects the lungs. Is called the pleural cavity. All right, so those three pressures are going to drive the inspiration and expiration of air in a very complicated manner, which you'll probably see, and you can look it up right now if you wanted to, but you'll get into that in anatomy if you do take it out. Hopefully you do. Now, in vertebrates, mammals being a type of vertebrate, what's going to happen here is the way in which CO2 and oxygen are going to be transported is by the use of hemoglobin. This is a large protein found inside of erythrocytes. Erythrocytes is another name for red blood cells. So these are red blood cells. Inside of them, there's about hundreds of thousands of hemoglobin molecules, and these hemoglobin molecules are attracted. They have affinity for oxygen and CO2. Okay, so as you breathe in oxygen, it binds on to hemoglobin. It's taken to the cells where it's unloaded. Yeah, so as oxygen is being unloaded to the cells are going to provide these erythrocytes with their CO2, right? So the CO2 that came out of metabolism. So cells are going to go ahead and release CO2 into hemoglobin to flow back to the respiratory system for exchange. So that process in and of itself has many functions. So it's not only transporting CO2 throughout the body, and it's not only gas exchange, it's also acid-base balance or pH balance in the blood, but also by formation of bicarbonate ions, which is essentially it's known as a buffer, okay, in the blood. So the way in which our blood pH is going to be kept at 7.35 to 7.45 is going to be by the use of an enzyme called carbonic anhydrase. So this carbonic anhydrase is going to go ahead and catalyze a reaction. It's an enzyme again. Catalyze a reaction between carbon dioxide and water inside of erythrocytes. And that right there is going to produce something called carbonic acid. So carbonic acid, H2CO3, is going to be broken down further into bicarbonate, HCO3, and hydrogen ions. Hydrogen ions are important in the because what they're going to do is they're going to go ahead and lower the pH of blood. Yeah, so by the simple formation of carbonic acid, this hydrogen ion can be hidden in the structure of a carbonic acid, which again is H2CO3. So if you add this hydrogen to this bicarbonate ion here, you make H2CO3, which is carbonic acid. In that manner, you can essentially hide many hydrogen ions in the blood into carbonic acid, which again is a buffering system of the blood to prevent your blood from becoming too acidic. Okay, because if there's too much hydrogen ion in the blood, free hydrogen ions, and then it's an acidic blood, which is very, very bad for your cells, as it can dissolve essentially membranes, and it's pretty dangerous. Alrighty, so last but not least for this lecture is animal nervous systems. So I'll give you an overview of the central nervous system, the brain spinal cord nerves, and I will call it a lecture. First and foremost, you should know that nervous systems, all nervous systems, are made up of neurons, and there's different types of neurons, and I'll get into them right now. So here's a generalized type of neuron. Okay, so neurons contain a cell body. So this is where all of the organelles are located: the nucleus, the ER, rough ER, smooth ER, Golgi. So again, cell body with all of its organelles. These are dendrites. Okay, these are dendrites which make connections to other neurons. This is called an axon right here, and this is the axon terminal. So as another neuron is connected to this neuron, it sends signals, electrical signals, through the dendrites. Okay, so just picture the axon terminal of another neuron being connected to the dendrite of this neuron. So those electrical signals are sent over through neurotransmitters, and those stimulate this neuron to essentially produce something called an action potential, which is an electrical impulse. So as that is produced here in the cell body, it is transmitted over to the end of the neuron through the axon. Okay, this is the axon right here. Once it reaches the axon terminal, then it produces neurotransmitters which go ahead and stimulate other neurons to do their job. Okay, so again, this whole neuron works by electrical gradients that are made by sodium and chloride for the most part, but also calcium as well. Alright, so all neurons work by the production of an action potential, which can be measured. An action potential is an electrical impulse. It can be measured, and it's actually broken down into different stages. And we're looking at the stages here. First is you have a resting potential. So at rest, neurons are not going to have a lot of activity. They will be at basal levels, meaning that they're going to be resting. Okay, so if they're resting, then their membranes are going to be anywhere from 60 to 80, negative 60 to negative 80 volts in millivolts in charge. Okay, that's the normal gradient for membranes. So as a neuron is being stimulated by an electrical impulse, that means that sodium is entering the cell body of this neuron. So if sodium enters, then enough sodium enters, then that causes a depolarization phase right here, which is a stimulation, an electrical stimulation of the neuron. So if that happens, then that means that the neuron is fully stimulated and it's ready to pass on the potential into the other neuron, and it does so by producing these molecules, these proteins called neurotransmitters. Okay, so you probably heard of a few neurotransmitters before. One example is serotonin, right? So that serotonin is very important in the brain. That's one neurotransmitter. So in order to make those neurotransmitters like serotonin, dopamine, GABA, those neurotransmitters, those neurons rather, need to come up to a certain voltage. All right, and that voltage is about positive 40. So they go from being negative 70 all the way to negative 65, anywhere from negative 60 to negative 80 for different cell types, especially neurons, all the way to positive 40, anywhere from positive 30 to positive 40. Okay, so that right there, if the neuron reaches a positive 40 millivolt charge, then that means that they are depolarizing. Okay, they're making that electrical impulse, which is forcing the neuron to produce neurotransmitters. So those neurotransmitters are made in the axon terminal, and then they're sent over to the next neurons to stimulate the same process in those neurons. So which is opening the sodium channels, allowing sodium to enter in the depolarization phase, and then allowing neurotransmitters to be made, be synthesized, their proteins, and then the same process happens again in the next neuron. Now, the neuron cannot stay depolarized. Okay, a neuron that stays depolarized causes very, very major problems in the brain. Okay, like epileptic seizures, for example, can take place. So what needs to happen is we need to calm that down. So we need to bring that neuron back to resting, and so that it's ready again to undergo the same process repeatedly. But again, you cannot leave it depolarized, otherwise it'll cause issues. So the second stage here is the repolarization phase. Okay, so in repolarizing, you want to bring that charge down from positive 40 all the way down to negative 60 to negative 80, depending on the cell type. So how are you going to do that? Well, you're going to open up potassium channels in your neuron, which are going to allow for the leakage of potassium to the outside of the cell. Okay, so why potassium? Well, potassium is positive. Okay, so if you kick potassium out of the cell, then the inside of the cell becomes less positive, ultimately going into the negative side. And they eventually reach a negative voltage here. And at that stage, that's called the hyperpolarization phase, which goes even lower than it's supposed to go. But that's just because sodium channels take a long time to close, so they're slower to close. So as they're closing, they're allowing more potassium to leave than they're supposed to. but not to worry, that's a normal adaptation, and the cell makes up for that by coming back to its normal voltage. So here's more or less what I mean. Here is your neuron. These neurons are lined by, you can't really see it here, let me go to another slide. Neurons are lined by membranes. Those membranes contain sodium potassium pumps, which allow for sodium to move in, potassium to move out, depending on what the cell is going to need. You can see them here a lot better. So this is basically showing you how the membrane of the neuron is studded with these sodium potassium pumps. So if sodium moves into the cell, then that causes a depolarization. If potassium moves, because the inside charge of the cell becomes a lot more positive. If potassium moves out of the cell, then that causes repolarization, because the inside of the cell becomes negative. All right, so you're releasing positive charge and thus making the charge go into the negative side. So what this is showing here is the propagation, the movement of an action potential, which begins here in the cell body, in the dendrites and cell body, and then it propagates over through the axon to the axon terminal, which is where neurotransmitters are going to be produced. Right? So a little bit more detail about the propagation of the action potential and the neuron itself. The entire axon is lined by these sheets. These are called myelin sheets. Okay, so here's one, here's another one. Myelin sheets are made up of Schwann cells, and these Schwann cells make protective fat layers that wrap around the axon here. That has several purposes, several functions there. One is protection. So the action potential, the electrical impulse that is transmitted from the cell body to the axon terminal cannot jump from one axon to a different axon. If that happens, then issues take place as well. Issues in the brain, so brain disorders can take place because of that. So these myelin sheets are going to prevent the jumping of action potentials from one axon to another different axon. That's one. Another function is to speed up the speed, if you will, right? The velocity of these action potentials can be sped up by these myelin sheets. And then a third function is potentiation. Yes, so in potentiating action potentials, what you do is you use these nodes of Rambier. So these nodes of Rambier are spaces in between the myelin sheets, which contain sodium channels. Okay, so if you allow sodium to enter these sodium channels, then that essentially potentiates, it makes a lot stronger that electrical impulse, which is needed so that it can reach optimally the next neuron. Once that action potential reaches the end of the neuron, which is called the axon terminal, then it goes ahead and stimulates the release of these vesicles. These vesicles are membrane sac or structures that carry neurotransmitters. Neurotransmitters are proteins, and they're chemicals essentially that make neurons work. Yeah, so they're activated by action potentials, electrical impulses. Once they get to the bottom of the neuron here, then they're released into the synapse, which is this space. This is called the synaptic cleft. And that synapse will provide the next neuron with its neurotransmitter and further its action potential. So what do neurotransmitters do? Well, they activate action potentials in neurons. Most neurotransmitters activate. There's others that don't activate. They actually inhibit neurons from carrying out with action potentials. Yeah, so they stop them. Okay, again, you don't want to keep neurons active all the time. You want to stop and have them rest for a little bit before the next one can be activated.
sites and cell body of a neuron may receive thousands of synaptic connections from other neurons. When these neurons are activated, they release neurotransmitters that open ligand-gated ion channels in the membrane of the receiving neuron. As ions flow through these channels, the membrane potential of the receiving neuron changes at the point of stimulus and passively spreads along the membrane. Some of these synapses shown here in green are considered excitatory because they make it more likely that the receiving neuron will fire an action potential. Excitatory signals depolarize the membrane, making the membrane potential less negative. This change is called an excitatory postsynaptic potential. Inhibitory signals, shown here coming from the red connections, hyperpolarize the membrane, making the membrane potential more negative and making the neuron less likely to fire an action potential. This change in the membrane potential is called an inhibitory postsynaptic potential. If the neuron receives enough excitatory stimulation, the membrane potential will depolarize sufficiently for the receiving neuron to fire an action potential, beginning in a region called the axon hillock. The changes in membrane potential of the axon hillock can be measured with a voltmeter. The voltmeter measures the difference between a recording electrode inside the cell and a reference electrode outside the cell. This value is normally about -70 millivolts, which is the resting potential of the neuron. If enough excitatory signals arrive close enough together in time, they may depolarize the membrane above a certain voltage, called the threshold potential. At this point, an action potential is generated. The action potential propagates the signal along the entire length of the axon. A look at the voltage-gated channels in the axon reveals how action potentials self-propagate down the axon. At the resting potential, the inside of the cell is negative relative to the outside of the cell. When a depolarizing potential reaches the axon hillock, where the voltage-gated channels begin in the axon, the inside of the cell in that region becomes less negative. The change in voltage triggers a shape change in the voltage-gated channels that causes the channels to open. Sodium channels are the first to open, and sodium ions enter the cell. The influx of positive charges further depolarizes the cell membrane and results in a positive moving spike in the membrane potential. The voltage-gated potassium channels open soon after the sodium channels and begin to release potassium ions outside the cell. As these positively charged potassium ions continue to leave the cell, the sodium channels begin to close, and the charge across the membrane in this area returns to its previous state. The voltmeter reading dips back toward the resting potential. A delay in the closing of the potassium channels causes a brief undershoot of the resting potential. The depolarized region is returned to normal as the sodium-potassium pump shuttles ions across the membrane, re-establishing the resting potential. Meanwhile, the changes in the membrane potential have opened adjacent sodium channels, and more sodium ions begin to enter farther down the membrane. The process continues in one direction, down the length of the axon. If this neuron makes an excitatory synapse with the next neuron, it too may fire an action potential.
Alright, so last point. These are the divisions of the nervous system. We have a central nervous system here and then a peripheral nervous system. So CNS and PNS. The central nervous system has several different divisions. So it's got an efferent division and then afferent division. The efferent division is concerned with motor action. So motor meaning movement. Okay, so motor is going to allow us to move. So for that, we're going to go ahead and utilize our muscles and our bones, evidently, right? Now the efferent division is going to be divided up into two smaller branches of the nervous system. One is the somatic nervous system, the other one is autonomic nervous system. The somatic nervous system we can control. Okay, so we're under the control of the somatic nervous system. which means that we can contract our muscles. So skeletal muscles that you actually think about moving, then again, that comprises the somatic nervous system. But then whatever you don't think about moving, you cannot control, then that would be the autonomic nervous system. So things like peristalsis, digestion, breathing, right? Anything that goes on inside of cells, in the gut, for example, small intestine, large intestine, you don't necessarily think about moving those. And the heart, then that's under the control of the autonomic nervous system, which divides into two further divisions here: parasympathetic and sympathetic. Okay, so another name for parasympathetic is rest and digest, and another name for sympathetic is the fight or flight response. Okay, so under the fight or flight response, then you're gonna be highly active. You're gonna be very, very active. A lot of metabolic actions are gonna take place here, and then sympathetic hormones are gonna govern the sympathetic branch of the nervous system. Okay, so hormones like epinephrine and norepinephrine, otherwise known as adrenaline, are going to go ahead and be secreted from the adrenal glands. That is also going to make thyroid hormones to be secreted, like T3 and T4. And those two right there are going to work indirectly with growth hormone, with insulin, glucagon, to be able to carry on with ATP production. The parasympathetic division is the opposite of sympathetic division. So when you're calm, you're not doing much, you're chilling, resting, then that's your parasympathetic system at work. Okay, so you're resting, but then at the same time, your digestion increases. So you don't want to be digesting too much under sympathetic division, right? And you don't want to be, very active under parasympathetic division. So they work in conjunction, but they have different actions. So here are the different levels of control of sympathetic and parasympathetic nerves. So sympathetic and parasympathetic impulses are sent down the spinal cord through the nerves over to the organs. All right, so the eyes, for example, the salivary glands in the oral cavity, the heart, the lungs, the stomach, the liver, the intestines, and then the genitals. All of it is under the control of both sympathetic and parasympathetic. So let me give you a few examples here. For example, under parasympathetic control, your heart is going to go ahead and increase in action, so heart rate is going to go up. Breathing is also going to increase, but then your secretions of acid are going to decrease, and your release of the bile for example, from the liver is going to decrease, because bile and stomach acid are digestive. So again, under sympathetic division, you do not want to be digesting too much food, because you're spending most of your energy moving. Now under parasympathetic control, then heart rate is going to go down, breathing is going to slow down, but your stomach acid secretion is going to increase, and your bile secretion is also going to increase. These two are digestive, yeah? So you're not very physically active per se, but you are active in digestion, if you will. All right, so brain divisions would be the last topic of the lecture here. So we have the human brain right here. This is a cross-section of the human brain, which divides up the brain into left and right. So we're looking at the right hemisphere of the brain here. We have several different general and large divisions of the brain. First is we're looking at the cerebrum. Okay, so this is the cerebrum right here. Right here we got the diencephalon, which includes the thalamus. Right here, the hypothalamus right there, and the epithalamus, which is located somewhere in this area, it's called the pineal gland. This is called the brainstem right here. The one in light green, that's the brainstem. Brainstem is divided up into three regions: the midbrain, the pons, and medulla oblongata. So behind, posterior to the brainstem, is the cerebellum, and those are the major divisions of the brain, of the central nervous system. Another structure of the CNS is the spinal cord, which moves down into the vertebrae located in your ventral, in your dorsal region, which is your back. So now let's talk about the cerebrum here real quick. The cerebrum is divided up into lobes, different lobes. Okay, so we're looking at the lobes here. In the front, right, by the forehead, you have your frontal lobe. This is the entire frontal lobe, which has major motor functions. It also allows us to carry on with cognition, thinking in higher level thinking, if you will. So complex decision making, multitasking, all of the major executive functions are governed by the frontal lobe, and it's mostly motor. Then we have the parietal lobe right here. The parietal lobe is basically the top of your head, and keep in mind we're looking at the brain in cross section. So keep in mind that for each one you're gonna have two, right? So for each lobe you're gonna have two sides. Here's the parietal lobe, which is involved in, it's got many different functions, which are mostly sensory. So these are mostly sensory functions. Sensory meaning it's processing information that's coming from the environment. Okay, so at the same time, it's talking to the motor, to the frontal lobe rather, which is working together with the parietal lobe in determining what needs to be done next, and dependent upon obviously the stimulus that came in. The occipital lobe here is located behind the back of your head. So the occipital lobe is mostly for visual stimulation. So both sensory and motor visual stimulation is controlled by the occipital lobe. Then you can't really see it here, but at least you can see a little bit of it. It's called the temporal lobe, which is right by the ears. Okay, so right behind the or deep to the ears rather. Keep in mind we have two sides, so two temporal lobes on each side, and it is involved in hearing. Okay, so each one of these lobes has a lot more divisions, hundreds of smaller divisions, which you can definitely go into. Unfortunately, we don't have a lot of time to go into every single division, but go ahead and do your research, and so you can find out further about each one of the divisions of these lobes. Again, the diencephalon located here contains the thalamus, which is the major relay station of the relay center rather of the brain. So everything incoming, everything coming from the nerves to the spinal cord into the brain has to be processed first by the thalamus, which is right there. So the thalamus is going to decide where to send the information. Yeah, so if it's visual information, for example, that's coming in from the eyes, which are somewhere around this region here, the eyes are connected to the thalamus via a optic chiasma, which has connections to the thalamus, and then from the thalamus it's connected back to the occipital lobe through an optic tract. So that's just one example out of the many functions that the thalamus has. The hypothalamus, which is located right below the thalamus, right around this area more or less, controls a lot of our primitive homeostatic functions. Also everything homeostasis, especially the production of hormones in the body, is governed and driven by the hypothalamus. Another part of the diencephalon is called the epithalamus. The epithalamus is also known as the pineal gland. The pineal gland produces a hormone called melatonin, which is inhibited by UV radiation. But as soon as night hits, then we start producing melatonin. It allows us to sleep. So it regulates something called circadian rhythms, which is essentially the day and night cycles. We are active during the day and we're resting during the night. The brainstem here, midbrain pons and medulla oblongata, that one has highly primitive functions. So functions in the body that are very important for survival, like breathing, respiration, heart rate, blood pressure. All of it is regulated by the brainstem here. And last but not least is the cerebellum. The cerebellum controls motor function. So if you move, and that's thanks to the cerebellum. Okay, so the cerebellum coordinates all skeletal muscle contraction. So if you have to move, if you have to run, for example, because a dog is chasing you, then as your nerves are receiving that input, right, they're transferring that input to the spinal cord, and then from the spinal cord into the cerebellum, which talks to the sensory part of your brain and the motor part of your brain. So those two are going to go ahead and integrate and coordinate information using evidently your eyes, because that stimulus came from the eyes, right? So these two parts integrate that information. They allow the brain to see that it's a pit bull running after you, and guess what? That's going to go ahead and talk again to the cerebellum, so that the cerebellum can make you essentially run, right, for your life if it's a real mad pit bull. So again, these are the divisions of the CNS. Again, very, very important for anatomy here.